Hello friends, my name is Arunya Bhatt and today we will discuss about microbial growth and cultivation of microbes in food. Introduction. Microbial growth in foods refers to the multiplication of microorganisms such as bacteria, yeast, and mold that can contaminate or spoil food. The growth of microorganisms in food can relate in food, spoil, reduce the nutritional value of food, and even pose a health risk to consume. Cultivation of microbes in food involves the use of specific conditions and methods to promote the growth and desired microorganisms such as probiotic or starter culture. These methods can be used to produce fermented food like yogurt, cheese, and surrogate to prevent the growth of harmful microorganisms in food. It is important to properly handle, store, and prepare food. This includes proper cleaning and sanitation of equipment and surface, controlling temperature and humidity, and ensuring proper storage condition. The use of food preservatives and food irradiation can also be used to prevent the growth and harmful microorganisms in food. Microbial growth in food. Microbial growth in food refer to the proliferations or multiplication of microorganisms such as bacteria, yeast, and mold that can contaminate or spoil food. The growth of these microorganisms in food can be influenced by various factors such as the temperature, moisture, pH, and presence of nutrients. If conditions are favorable, microorganisms can rapidly multiply in food, leading to spoilage, degradation, and potential health risk to consume who consume for consumer who consume the contaminated food. Bacteria growth curve. The bacterial growth curve is a graphical representation of population growth of bacteria over time in culture medium. It is a typical S-shaped curve that has four distinct phases. First phase is lag phase. It is also known as adaptation phase. In this phase, maximum nutrition is present, no cell division, no increase in number of cells, increase in size of bacteria. Duration of lag phase varies according to the conditions and species of bacteria. Then second phase is log phase or exponential phase. It is also known as log phase. Maximum growth of microorganism in this phase. Maximum substrate utilization in this phase. Rapid growth of bacteria take place. Maximum specific growth rate. Then stationary phase. In this phase, number of death is equal to number of growth. There is no increase in number of bacteria. Deficiency of nutrient started. This phase maintained for hour or a day. Death phase or decline phase. In this, microorganisms start die due to the release of toxin, deficiency of nutrient, and accumulation of waste product in this phase. Factor affecting microbes in food. Food of plant and animal origin play an important role in microbial growth. The ability of microorganisms to grow or multiply in such food depend on the food environment. There are two factors that affect the growth of microorganisms food. First, intrinsic factor. Second is extrinsic factor. In intrinsic factor, pH, water activity, nutrient content, antimicrobial substance, and in extrinsic factor, temperature of storage, relative humidity, atmosphere, light, processing, and storage condition. In intrinsic factor, the internal self parameter of plant and animal tissue are referred to as intrinsic factor. These factor includes pH, which is also known which which full form is hydrogen ion concentration. The molds have pH. The acidity and alkalinity of the food can affect the growth of microorganisms. Most bacteria grow well in slightly acidic environment between pH 4.6 to 7.5. Some example are molds have pH 1.5 to 9.0, yeast have pH 2.0 to 8.5, gram positive have 4.0 to 8.5, gram negative have 4.5 to 9.0. Based on pH range, microorganisms can be grouped as neutrophiles, acidophile, alkalophile. pH. Then second is water activity. Is the water activity is the amount of water available for biological function, which can be reduced by an osmotic effect. Water activity Water activity of food product is measure of the availability of free water that can support microbial growth. Food with low water activity, like dried food, it has it, they cannot support the microbial growth, but which are fresh food support the growth of microorganisms. Second, third is nutrient content. The amount and type of nutrient in food product can affect the growth of microorganisms. Foods rich in protein and carbohydrate are more prone to bacterial growth than those with low nutrient content. Antimicrobial substances. Some food contain natural antimicrobial substances such as spices and herbs that can inhibit microbial growth. Extrinsic factor. These are external factors that can affect microbial growth and survival in food, and they include temperature, relative humidity, atmosphere, light, processing and storage temperature. The enzyme. The temperature at which food is stored and handled can affect the growth and survival of microorganisms. Most bacteria grow rapidly in the temperature range of 4 degrees Celsius to 60 degrees Celsius. Based on some temperature, some bacteria are psychrotrophs, mesophiles, thermophiles. Relative humidity. The humidity level in the environment can affect the water activity of the food, its ability to support microbial growth. Atmosphere. The atmosphere in which the food is stored can affect microbial growth. Some microorganisms require oxygen to grow, while other grow in the absence of oxygen. Light. Exposure to light can affect the growth of certain microorganisms in food processing and storage condition. The condition under which food is processed, packaged and stored can affect the growth and survival of microorganisms. Proper handling and storage condition can help prevent the growth of harmful microorganisms and ensure food safety. Cultivation of microorganisms in the process of growing and propagating microorganisms in a controlled environment such as laboratory or industrial setting. The goal of cultivation microorganisms is to obtain pure culture of specific microorganisms for scientific, medical or commercial purpose. Here are the general steps involved in the cultivation of microorganisms. First, inoculation. The first step in cultivating microorganisms is to obtain a sample of the microorganism and transfer it to a sterile growth medium. This can be done by swabbing a surface, taking a sample of soil, or collecting a sample from liquid culture. Incubation. After inoculation, the growth medium is incubated under conditions that are optimal For the growth of the microorganism, this typically involves maintaining a specific temperature, humidity, and pH. Third, isolation. Once the microorganism have grown, they can be isolated into pure culture. This can be done using various techniques such as stick plating or spread plating or pour plating, which which involves spreading the microorganism over the surface of the growth medium to obtain isolated colonies. Identification. Once isolated, the microbes can be identified using various techniques such as microscopy, biochemical tests, or DNA sequencing. In addition to these general studies are various specialized techniques and growth media that can be used to cultivate specific type of microorganisms. For example, some microorganisms require specific nutrient, oxygen level, and growth condition to grow, and specialized growth medium can be used to support their growth. The cultivation of microorganisms requires careful attention to sterile technique, as contamination can lead to the growth of unwanted microorganisms and compromise the purity of the culture. Purpose of cultivation. Cultivation of microorganisms in food is essential for several reasons, including quality control. Cultivating microorganisms in food can help monitor the quality and safety of food product. The presence of certain microorganisms food can cause spoilage or pose a health risk to consumer. By culturing and identifying the microorganisms, food manufacturing can take measures to control and prevent their growth. Fermentation. Second, fermentation. Many foods such as yogurt, cheese, sauerkraut are produced through fermentation by a specific strain of microorganisms. Cultivating and maintaining these microorganisms is crucial for the production of consistent, high-quality food product. Third, food preservation. Microbial culture can be used to preserve food by inhibiting the growth of spoilage and pathogenic microorganisms. For example, lactic acid bacteria are commonly used in the production of fermented vegetable to prevent the growth and harmful bacteria. Nutritional value. Some microorganisms, such as certain strain of probiotics, can have a beneficial effect on human health. Cultivating and incorporating these microorganisms into food products can enhance their nutritional value and promote health. Pure culture technique. Pure culture techniques are used to isolate and grow a single species of microorganism from a mixed population. The primary goal of pure culture technique is to obtain a culture that is free from any other species and contaminants. There are several techniques used to obtain pure culture of microorganisms, including first, stick plate method. This technique involves spreading a small amount of a mixed culture onto an agar plate using an inoculation loop. The loop is then used to spread the bacteria in a series of streaks, with each subsequent streak containing fewer bacteria than the previous one. Fewer bacteria than previous one. This result in isolated colonies of bacteria that can be further isolated and grown into pure culture. Pour plate culture. In this method, a small amount of mixed culture is added to a sterile petri dish and then melted agar is poured over it. The mixture is then allowed to solidify and colonies of bacteria grow within the agar. As the colonies grow, they become physically separated, allowing for individual colonies to be isolated and grown into pure culture. Spread plate method. In this method, a small amount of mixed culture is spread evenly onto the surface of an agar plate using a sterile spreader. The bacteria grow within the agar, and individual colonies can be isolated and grown into pure culture. So, thank you.